Yeah, so welcome everybody. Today I'm actually replacing the talk of Camille Oludak, who was not able to make it today. I want to focus today on quality control of functional MRI data that I acquired at ultra-high field, mainly on 7 Tesla. So basically the 7 Tesla fMRI is providing us with novel opportunities with respect to SNR, but also with respect to the resolution that you're able to acquire with those systems. So basically with 7 Tesla fMRI, it's nowadays possible to indeed acquire functional images like 2-2 star rated images in the order of the sub-millimeter range. For example, 0.75 millimeters or 0.5 millimeters can be used for actually tracking cognitive tasks. And this is, of course, giving us a lot of more opportunities with respect to the hypothesis we can test, with respect to the scale of the analysis that we can perform. And this is just one study I want to show you today, because this is a study that has been used a lot also for validating some of the analysis technologies. So here what was done is, in this paper, there was a focus on the primary motor cortex. It was just a very small step acquired, and basically participants were asked to either tap the hand so to move and tap the hand, providing motor output and sensory input, to only move the hand without touching, to get only receive passive touch, or to do ipsilateral tapping. And this is actually providing you with very nice input, output, or combined input and output data. And this was actually then used for analyzing layer-specific analysis profiles. And what you can see is there's a very nice biphasic response here with peaks in the upper and the lower layers for the combined motor output and sensory input condition. This is a very nice paper showing that in principle you can use those sub-millimeter resolution data to really acquire layer-specific signal change that you can then analyze with respect to, for example, distracting input from output signal flows. And this is just a very brief overview over the possibilities you have with those layer-specific data. So, for example, dissociating input from output signal flows is just one possibility. Another possibility is, for example, to look at feed-forward versus feedback processes, which also often take place in different cortical depths. There's also the possibility to look more closely into processes of perceptual sharpening, or processes that are associated with associative cognitive function. So it's just a lot of possibilities that you have in order to actually really describe the cortex in a third dimension, in the dimension of the cortical depth. And this is actually opening up a really novel area of fascinating questions. And this is why today I want to actually excite you a little bit, to jump into this novel field of 7 Tesla fMRI on the one hand, but on the other hand I also want to draw attention to a lot of artifacts that actually are caused by those kind of data, and you have to be very, very careful actually controlling those. The purpose of the talk today is therefore to provide an overview over the most critical 7 Tesla fMRI data artifacts, and I want to focus actually on those that you at the moment can to a certain extent handle using available software tools. So I'm actually trying to give you a small guideline also how to handle some of those artifacts using available software tools. An artifact, as I define it here, is, as we heard before, either something that is observed but not actually present, but actually particularly in layer-specific analysis or in looking at fMRI signals at the sub-millimeter range, you also may actually observe something that is there, but it may actually lead you to draw wrong conclusions if you don't interpret the data correctly. So it's those kind of two types of artifacts that I want to focus on today. So I want to focus on signal dropouts, geometric distortions, motion, and also changes in the B0 field, layer segmentation, and vein effects. So let's just start with the signal dropouts. Signal dropouts can be a quite serious concern, particularly when you look at subscale functional imaging data. So this study here used 2-photon microscopy in rats and mice to actually see how the bold signal is dependent on the orientation of the cortical surface to the B0 magnetic field. And here what the authors did was modeling the contribution of the cortical surface with respect to the static magnetic field. Here you see this phi z angle that is actually showing you that there's a very different orientation of the different areas of the cortex with respect to this magnetic field here just shown in this vector. And you see that there is a strong dependence of the bold signal dependent on the orientation of the cortical surface to the B0 magnetic field. It can be a variation up to 40%, which is quite dramatic, I think. And this is has also been shown using real fMRI data in human subjects. So you do have this very strong influence of the orientation of the cortex with respect to the signal you actually measure, and this can, for example, lead to dropouts. If you see here those layer-specific effects I showed you before, they can really sometimes disappear in those very highly folded areas of the cortex, which can sometimes be corrected, as I show you at the next slide. So there's the different possibilities you have to actually model the contribution of the curvature or the on your bold signal. One possibility is described in this paper here. So what you can actually do is, for example, using a cortical surface reconstruction, you can model for each vertex, you can actually model the respective angle distribution that you get, and then you use it as a regressor in your model. So, for example, here it is shown that you can see that area 4 and area 2 are compared, and you see a lot more of those signal variations in motor cortex compared to the sensory cortex. So the better the delta R2 star is just showing you the difference between the 0 vector angle and the 90 degrees vector angle. And you see here, for example, in this study, using this as a regressor, that there's also a high correspondence to your FA values with respect to this parameter. Another possibility you have is a very different one. So you can also think about your sequence. So as you also saw in the previous slide, there is a particular problem in gradient echo sequences with respect to this particular artifact. And there is a lot of studies that show that there's less sensitivity towards signal dropouts for spin echo sequences, for example, because they use this second, so they actually have 2 RF pulses, and the second refocusing pulse is then to a certain extent reducing the influence of the orientation on the signal. So this is also what you see here. So spin echo sequences do have the problem of being a bit slower compared to the gradient echo, but it's still a problem, it's still a possibility, for example, for very robust block design experiments. The second aspect I want to talk about is geometric distortions, which actually pose a serious problem particularly for API data. So as you also heard before in the first talk, you can actually do those contour lines when you look at how an anatomical image and a functional image can be realigned to each other. So very often what you do, particularly in 7 Tesla fMRI studies or 7 Tesla MRI studies, is that you have a very high resolution anatomical image, which is very often giving you the subtle details of the anatomical structure you're looking at, and then you're actually acquiring an additional functional image where you then want to actually relate those data to. A particular problem that you're then facing is if the functional image is not 100% correctly overlaid to the contours of the structural image, then of course you can actually not really interpret the data anymore, particularly not at the really sub-millimeter scale. So this problem is a major problem, particularly in high field analysis, because you do want to have this really sub-millimeter precision in your data, which is actually sometimes not possible when the data are distorted, even after you apply, for example, affine registration to the image. And one possibility we have, and that we also use at our site in Magdeburg, is to implement point spread function mapping into the acquisition pipeline. So what is done here is actually there is a reference scan measured directly before you measure your API sequence. And this reference scan is then used for applying a 3D Fourier transformation, and is giving you then this 3D PSF image, which is basically calculating the local shift in the distorted phase encoding dimension, creating a shift map that can then actually be used to correct your acquired API data sets. And this correction is actually done online. So you actually, at the output level, you already get those distorted and corrected data out of your processing pipeline. And when you look at one example image here, so those PSF mapping correction can actually massively reduce those geometric distortions. So here you again have the contour line of the reference image and the acquired API image with the correction applied and without the correction applied, and you see that those two images actually strongly realign again. So there's a massive improvement by this specific PSF correction. This is another example where the influence of the different phase encoding directions for API images is shown. And again, you do see actually that the corrected image is much less distorted, much less changing with respect to the face encoding compared to the uncorrected image. And here it's also, as you heard in the first talk, we just did a subtraction image, and you actually see that there is a lot of difference in the signal for the non-corrected image, but there is a relatively stable signal for the PSF corrected image. However, this is a methodology which I should point out here, which has to be implemented into your sequence. So it's not something that you can do post-hoc with your data, but it's actually really something that the physicist team is implementing into your MRI sequence. Another possibility you have actually to correct for geometric distortions, or let's say to handle geometric distortions, is to use anatomical data with identical readout as functional data. It's actually a different approach. So rather than actually correcting the geometric distortions that you have in your image, the approach taken here is that you actually acquire an anatomical image that has the same, let's say, the same distortions as your functional image, and then those two images are then aligned and can also be interpreted in the same space. So here in that example, this is an MI API sequence that has the same readout as the functional data. And then actually this can be used, for example, for free surfer segmentation. So you can actually put this simulated MP-RAGE image into your anatomical free surfer pipeline, and can then also conduct surface mapping. And this framework that you have there is then actually presenting with the same distortions as the functional image. So when you register the data, for example, you're actually analyzing them in the same space. Another problem that we heard about before is motion. Motion is also a major problem, of course, in 3 Tesla fMRI experiments. It becomes, however, extremely critical for 7 Tesla fMRI experiments because you sometimes, as I showed at the beginning, you are looking at voxel sizes of around 0.5 or 0.75 millimeters. So even a motion of around 1 millimeter is getting you into serious trouble if you want to look at layer-specific analysis, if you want to conduct layer-specific analysis. And also another problem that you have with 7 Tesla fMRI data is that you often acquire SLAP data. So you often acquire data with a reduced field of view to actually really improve the SNR and the signal change within your small SLAP. And what can then happen is, I think it was a very interesting paper that was published at our site 2 years ago, is that particularly for those SLAP data, you do often have very erroneous retrospective motion correction. So what we saw here is that the correlation coefficient between the correct image and the retrospectively motion corrected image, for example, using SPM, was very, very weak. And this was actually because the signal fluctuation that was induced by the experimental paradigm was massively, of course, influencing the data. So you can imagine this was a SLAP acquired in the primary visual cortex. And then actually conducting a visually driven experiment is changing almost the whole signal intensity within your SLAP. And this is actually taken then by the motion correction algorithm as a change in the voxel location. So the algorithm can no longer actually detect doesn't anymore have any fixed locations to focus on. So the whole image was shifted according to the experimental manipulation. So it was actually not working with respect to the standard analysis tools. And another problem, of course, that you have is not only that with small SLAP data of high signal intensity changes that the motion itself cannot be corrected, but of course you do also have strong effects of the change of position within this motion that actually then also leads to distortions with respect to gradient field nonlinearities. And one possibility that you have to correct for those kind of motion effects, some of those motion artifacts, is to use prospective motion technologies. Again, as before, this is a technology that has to be implemented into the MR environment, so you don't apply it post-hoc, but you have to really build it into the system. And this PMC correction technique is composed of an optical camera with included lighting that tracks the motion of a passive MRI phase tracker. This is a very small device, and this is actually how it looks in the system. So you actually have this camera attached to the scanner bore in around, I think, ideally 20 centimeters away from the target. And the target then is actually tracking the motion of your brain, in that case, online. And this is why you usually fix it to a mouthpiece that is then attached to the upper teeth, because they really move a lot in line of, they're moving in line with your head motion. Some sites, I think, do also attach this to the forefront, to the skin. However, you have to remember that you can also move your skin without moving your head. So I think attaching it to the mouthpiece is giving you much more reliable results. The camera can then track 6 degrees of freedom, the motion in 6 degrees of freedom. This is just an example of the tracking data, which are highly accurate. So they have an accuracy of around 0.01 millimeters. And they're as accurate that they can even detect single heartbeats that make your brain pulsate, actually. So they're highly accurate. And what then happens is that those data are tracked online, and the field of view is updated online to actually correct for those head motions. And this is one example image here where a very high resolution 0.25 image was acquired with and without such prospective motion correction technologies, and you see a massive improvement when the correction is on. The only thing that you have to have in mind or keep in mind is when you want to use those technologies is that you have to calibrate it before use. So at the beginning of the day, you calibrate the technique, and then you can use it for multiple subjects, multiple scans afterwards. Oh, sorry, and I also wanted to say that you can also combine this prospective motion correction with additional technologies that correct for the gradient nonlinearities. So there's a lot of possibilities also to extend this algorithm further to also correct for the distortion that are associated with the motion, as I indicated before. A further, I think, important aspect about the quality control of high-resolution data is to think about how you actually finally segment your tissue, because mostly when you do 7 Tesla imaging, you actually do want to have sub-millimeter and very accurate segmentation of the different tissues involved. And this is here one example of an algorithm that was developed to automatically segment biologically based cortical layers in the big brain. However, this technology is so far only available really for data where also the sub-millimeter tissue components are available. So this is at the moment not available for in vivo data, but only for ex vivo data. So for in vivo data, actually you do have still to model the layers, so you cannot directly measure them, but you just have to rely on certain modeling technologies that are available. And this paper, which I think is very nice, compared the different technologies to model cortical layers and validated that the equivolume model seems to be most correct for actually estimating the layers in different cortical depths. So just very briefly, the main assumption of the equivolume model is that the changes of the surfaces across different cortical depths and across different curvatures are actually preserving the volume, and however are then changing the cortical thickness but also the spread of the layers. And this seems to be most closely to the real biologically based data. And therefore, implementing this algorithm into your pipeline is actually giving you the possibility, rather than just dividing, the cortical depth into equidistant layers, it's giving you a more accurate estimation of how the folding influences your layer estimations. And this algorithm is implemented in the MyPath framework, for example, but it has recently also been implemented into the FreeSurfer pipeline. So you can also use this in FreeSurfer to more accurately actually model the contribution of the depth on the layer segmentation. The last aspect I want to focus on is the effect of veins on your data. So I think it's a major issue in 7 Tesla fMRI that the vasculature is strongly influencing your ball signal change, and this is particularly important if you want to look at cortical depth specific changes in your signal. So the bold API effect is mostly driven by venous veins, but also venules, and the major problem is, for one, that the ascending veins are not equally distributed across different cortical depths. So actually, even when you have an equal neuronal signal across all cortical layers, you will have actually a higher signal at the superficial layers, just because of the contribution of the distribution of the veins in those different cortical depths. So your pure signal is actually not wrong in the sense of, you remember the second definition of an artifact. So this is actually a signal that is true, it's there, but you may not be able to derive correct conclusions, because it may not actually indicate you a strong activation of your superficial layers. It just may indicate to you that the venous contribution is higher. So this is a major problem of those draining veins. A second problem is that you have those pile veins that just spread across the cortical surface, and actually they spread around your signal a lot superficially to the perpendicular to the cortical surface. And it can also make your both signal then very, very imprecise. And there's actually a couple of technologies that are trying to correct for those kind of problems. One, perhaps the simplest approach, is you can try to remove pile veins, either by creating a filter and filtering out just the both signal that is derived by those venous compartments, or you can also just remove the upper 20% of the cortex. So this is a very nice study here where a stimulus was used, that is a visual stimulation protocol was used that is producing like an M shape in the visual field. And you see here that it's particularly error-prone in the superficial layers, whereas the signal is much more precise in the lower and middle cortical layers. So just by removing the upper part of the cortex and focusing on the lower layers is giving you a more precise signal just for a first start. However, there are also more sophisticated technologies to correct for those kind of problems. So what you can, for example, do is you can combine your functional task with a guess challenge, where you then actually model the contribution of just the baseline signal fluctuations. So here, for example, it's a very nice example of, again, focusing on primary motor cortex and a tapping paradigm, as I introduced at the beginning. And here, when you just look at the raw signal change that is driven by the task, you may think that your activation peak is actually in the superficial cortical layers. However, what you see here, just by looking at the data that are produced by the GUST challenge, just during pure rest, is giving a very, very similar profile. And just by actually normalizing your functional data by those resting data, it's actually then showing you that your true activation peak is actually at the middle of the cortical depth. This is also similar when you use a normalized cortical profile. So this is technologies that you can use for actually correcting your data for those baseline venous compartments, and then trying to find out which is really the effect of your experimental manipulation on your data. Another possibility that you have when you want to focus on connectivity, layer-dependent connectivity, for example, is that you can model the influence of different compartments on your dynamic causal modeling approach. So here, what was basically done is it was a little bit of a simplified approach of modeling only two compartments. So it was assumed that there was just two different compartments in cortical depth. And there was then a factor introduced that was modeling the draining time that the blood takes from lower to superficial layers with respect to the signal influence of the draining veins. And the coupling parameter was modeled that shows you the strength of this influence. And it's just this one additional parameter that was added actually to the DCM model, which can then allow you to test hypotheses, for example, on how the lower layers are influencing the upper layers. And with respect to such models, which is actually, I think, a strong developmental procedure at the moment to actually develop further those models. This is a very, very new approach that has just been uploaded in bioarchives. So here, actually, this approach is taken further, but here, in that case, the effect of ascending veins and venules, but also draining veins at the surface, is modeled individually. So here, venules are modeled according to the balloon model, which we also, for example, use in SPM, but it's here expanded for multiple cortical depths. And here, in that approach, the ascending veins are modeled as the sum of blood inflows from the venules of the same depth, and the lower depth of the ascending vein minus the outflow of the upper depth. So it's a little bit complicated, also a lot of mathematics involved. What I just want to show you here today is that this model is actually really trying to model the contributions of each of those different blood vessels on your data, which is then, for example, allowing you also to test different assumptions, but also to validate your data. So here, for example, this model was used to reproduce the functional activity profile that I showed to you at the first slide. So here, this is again this biphasic response that is driven by touch and motor output, as I showed you at the beginning, using CBV and both signals. And this model was actually able to reproduce exactly this biphasic response for the CBV response, but the actually one peak response for the Bolt response. So it's just this kind of modeling technologies that you can use also for verify your analysis, but also to derive specific hypotheses of how your experimental paradigm can perhaps drive your real measured data. And this is already, so I'm already coming to the end of my talk. So I think one very important thing that you have to keep in mind is that successful 7 Tesla fMRI studies is not possible without those kind of quality controls, because the raw data are often very highly distorted, and also the data that you get out of your pipeline are often not really interpretable in the way you want to interpret them if you don't correct for those kind of artifacts. And those tools, as you see, are constantly improved and updated. So it's a very new field. 7 Tesla fMRI is just developing, and it's actually a lot of people who are working right now on those methods. So it's really worth to getting in contact with those people and actually building up a very collaborative and multidisciplinary network to actually always trying to keep updated on the best ways to correct your artifacts. So in a way, there's not yet a kind of a standard way to correct those data. It's really something that the researchers are still developing and improving. And as I want to point out to you, in case you are planning to do some 7 Tesla experiments, and you, for example, are thinking about choosing your site for doing so, it is important that some of those methods have to be implemented into the MR scanner environment. So it is worth actually communicating with the site you want to do your research, whether those technologies such as prospective motion correction or PSF mapping are already implemented, are in place, and can be used by you in your study. And with this, I would like to thank you very much for your attention.